There are many kinds of viruses. Just to give you a flavor of how weird these things are, that all living cells have double-stranded DNA genomes. And the human genome is 3 billion base pairs of double-stranded DNA. That is DS, double-stranded DNA. Some example viruses, genome, and then capsid, because it's kind of a weird world out there. Smallpox it's about 200,000 base pairs of double-stranded DNA. So the smallpox genome is made of the same kind of stuff that our genome is made of, double-stranded DNA. Its capsid is protein and lipid. Rhinovirus, which was probably that sneeze right there, that's the common cold, or that cough, is about 7,000 nucleotides of single-stranded RNA. And it's capsided protein only. So there's an RNA genome, which is pretty weird. Hepatitis B is about 3,300 base pairs, or nucleotides, of single-stranded DNA. And that's protein plus lipid. And the last example is rotavirus. This causes infant diarrhea. And if you have kids, you know what this is a sort of characteristic smell. It's pretty bad news. They have about 10 chromosomes. It's not fatal. It's just rather unpleasant. Each about 1,000 base pairs long of double-stranded RNA. And it's got a protein capsid. So what's the point of showing all of these things? You can have an RNA genome, it can be double-stranded, it can be single-stranded. You can have a DNA genome, it can be double-stranded, it can be single-stranded. The virus world is a very interesting and weird world. The world of every other living cell is very simple. Double-stranded DNA, that's it. Yeah, Levi. dsDNA, double-stranded DNA. It's just an abbreviation. Other questions? And there's no correlation between the kind of genome they've got and how nasty they are. It's just, that also suggests lots of independent evolutions of these things, that they probably have different origins in evolutionary history because they're all so very different from one another. Yeah? So the question is, so why do the genome sizes vary so much? It depends on, because that gives you a whole idea of how many genes they have. Why is smallpox so big? It isn't clear whether all the stuff in the smallpox genome is necessary or not. Because you can certainly get by with little itty-bitty genomes, because you don't need many genes. In fact, I should say something about that in a second. But yeah, it's not well known, all of the details. And there's a lot of interesting research on that sort of stuff. So the minimal virus genome, how can they get away with a genome that's 1 million times smaller than ours? And the answer is you've got to have a gene for the capsid protein. What do you have to have for the very minimum to be a virus? 2, the gene for a replicating enzyme if your genome isn't double-stranded DNA. If you're double-stranded DNA, then the host DNA polymerase can take care of it just fine. You need genes for proteins to release virus from the cell and for genes to suppress host gene. Optional. So there's some genuinely fiendish viruses that not only do they inject their genome and have their proteins and the like made, but there are some of them that actually actively shut off host promoters and turn on their own. So not only does the host make copies of the virus, but they see to it that the host makes nothing that it ought to. So not only are you making many copies of the virus, but you're not doing anything else. It's extremely lethal for the cell, very good for the virus, extremely aggressive. There's a whole bunch of other viruses have evolved other proteins for subverting all sorts of host defenses because, of course, any virus that picks up a protein like that is going to be at an advantage. But the minimum thing is you got to make the capsid, because that's not something that comes naturally to the host. You have to copy your genome. Now, you can maybe use the host DNA polymerase, but if you got some funky, weird RNA genome, you're going to have to have an enzyme for that. And probably need some proteins to help you get out of the cell, rather than having to wait for the cell to burst. And you might want to shut the host genes down, but that's optional. So that's why they can get away with genomes that might have 2 or 3 or 5 genes at the most. That's all you really need to just get yourself copied. They're sort of the most stripped down organisms that exist. Questions about those? We'll talk about one other class of viruses. Okay. There's one specific class of viruses that is particularly interesting to talk about, which are called retroviruses. For example, HIV, which causes AIDS, and feline leukemia virus, and others. There are a number of viruses which are retroviruses. And I'll talk about why retro in a second, what that means. The reason why we care about them, if they've come to be known, is because for a long time no one cared about them because they were sort of obscure, weird kind of viruses. They didn't typically infect humans. Turns out HIV is a retrovirus. So it went from a laboratory curiosity to a very serious issue in the 1980s and 1990s. Also, if you have a cat, you may have got them vaccinated for feline leukemia virus, which is also a retrovirus. So they have a particularly interesting life cycle that I will talk about. I'll show you an animation in a sec. They have a single-stranded RNA genome, right? Which means they cannot be copied. They can't replicate using the host's DNA polymerase. Just can't. DNA polymerase only makes copies of DNA molecules. There are some special features that they have. That the virus makes a DNA copy of the RNA genome. This is the retro part. I'll explain that in a second. And then virus inserts copy into the host's genome. Therefore, the virus is passed to the daughter cells, which makes it particularly insidious. And I'll talk about that in a second. But what makes this retro, normally, the central dogma, DNA, RNA, protein. That's where information flows. This is making a DNA copy of RNA. That's going backwards. So normally, DNA to RNA to protein. The retro does this. It goes backwards from RNA to DNA. So that's why it's called a retrovirus. It does central dogma backwards. In fact, when this first was proposed, people said, nah, it can't work that way. In fact, the original people who proposed it were treated as lunatics, because, well, not lunatics, but they were like, you guys can't be serious. You must have done your experiments wrong, because it goes DNA, RNA, protein, pal. That's how it works. And it turns out actually sometimes it goes the other way. So if you turn your attention to the next page of the information packed handout, actually somebody's a cool Twitter question. They asked, does the virus replicate by meiosis? And the answer is it's actually neither. It's not mitosis nor meiosis. It's its own viral way. There's no formal mitosis or meiosis in either of those guys. It doesn't work quite the same as a regular cell. So if you look at the next page of today's information packed handout, this is the HIV life cycle. So there's a couple things about HIV that are known in more detail. There's a capsid inside, and there's this glycoprotein. It has a name, which is worth knowing because you'll hear about it from time to time. It's called GP120. It's labeled in today's handout. There's an envelope of lipid, and then there's a protein coat inside. It's a multi-layer kind of structure. And then there's the RNA genome, and then there's this enzyme called reverse transcriptase, which is really important. And we'll talk about that in a second. How does this guy work? Like all viruses, let me skip ahead through this blah, blah, blah part. Got your classic host cell, nucleus, DNA in the nucleus. HIV comes along, and it only binds to some cells. If you look at it's step 1 on the chart in front of you. It's known, because HIV is such a serious thing, with such a lethal disease, it's been studied in extreme detail. GP120 on the virus binds to a protein called CD4 on the surface of the cell. And CD4 is an essential protein for certain cells in the immune system. Only some cells have it. Certain cells in the immune system, certain brain cells, other ones. So that's why HIV infects only some cells. And the GP120 on here binds to the CD4 there. You may have heard of people who are immune to HIV. It turns out it actually GP120 binds to CD4 and another sort of accessory protein. Those people have a mutation in that accessory protein, so HIV cannot bind to that. And they are resistant to HIV because they lack its target. It does not infect their cells. And they are, if not impossible, extremely difficult to infect with HIV because there is not that specific recognition. So then, like any other virus, if it gets recognized, it goes into the cytoplasm. And then here's the weird step. This is where the retro comes in. That yellow thing is reverse transcriptase. Reverse transcriptase. What's transcription? What process does it do? It copies what to what? DNA to RNA. So what would reverse transcription be? RNA to DNA. That's exactly what this guy does. So it makes a DNA copy. It reads RNA and makes DNA. It's that heretical enzyme that goes backwards. That's the reverse. That's the retro part, the reverse transcription part. Makes a copy of the DNA genome. And this is a particularly cool enzyme because not only can it copy one strand of DNA, but it can also copy the second. So you get double-stranded DNA. It's a rather unusual enzyme in that respect. It catalyzes 2 different reactions. And then that double-stranded DNA, that's the virus's genome, goes into the nucleus. And there's another enzyme that actually cuts the host's DNA and inserts the virus's DNA. So now this is in some cells this is a particularly insidious thing. Let me say the following: if this cell divides, or the virus now enters what's a latent period. It may sit like this without making any viruses for quite some time, between 1 and 10 years, or between 0 and 10 years. Why is this bad news? Because what it means is that if this cell divides, the daughter cells will have the virus in them. So now we're having many more potential virus factors. The other thing is, this is how it evades your immune system. Your immune system checks every cell in your body for what proteins it's making. If it's making an inappropriate protein, like a virus protein, then your immune system kills that cell. It says, I don't care, that might be an important cell, but it's about to make a virus. I'm going to take it out of the picture so I won't get infected. If it's not making anything nasty, then nobody knows. Your immune system doesn't know that this is a sort of a ticking time bomb. And so this cell can be latent in your body for many, many, many years before finally it goes on and makes the virus, as we'll see in a second. The other thing that makes this insidious is that if you think about a virus strategy, If I'm a virus and I want to infect the largest number of people, I don't want to kill my host right away. Because if I kill him right away, he drops dead and doesn't spread it to anybody. I want my victims to be infectious and active for a long time. That's why the common cold does so well. Because you get sick and you come to Lipke and you spread it to zillions of people. If it was a really virulent virus, you'd be flat on your back not infecting anyone. So that would not be a useful strategy for that virus. Right? HIV is particularly nasty because you can be infectious and sexually active for 10 or 15 years before you get too sick to spread the virus. So you can have all those sex partners for all those years if people are doing unprotected sex will get infected. So it's particularly nasty. And that comes from this business that it can stay latent. Right? So that's one. And then the next thing that happens, at some point, it's not quite clear what the trigger is, the virus gets activated. The pro-virus gets activated. And its promoter gets turned on, and it makes an RNA, because cell doesn't know any better. Those RNAs get translated, and they make viral proteins. And then those viral proteins assemble, and you get virus and all that. There's a further wrinkle to this. If you look at step 11, the proteins are the HIV has all sorts of weird heretical things in it. The proteins are actually synthesized as one monster protein. And there's an enzyme within that called HIV protease that actually snips it at several different places to cut it up into individual functional units. So there's one monster protein that has that reverse transcriptase, protease, this, that, and the other thing. The protease comes along and snips it into several different smaller pieces, each of which is active. So it synthesizes a monster protein snipped down, which is a very unusual thing, but how this works. All of these unusual things, of course, are targets for therapy, which we'll talk about next time. So then this cell, and with HIV, typically the cell doesn't die. It keeps producing virus for budding off for a long period of time. Eventually, it does eventually die, but it doesn't, I should say carefully, it eventually dies, but it doesn't burst as much as it keeps sort of budding off more and more and more virus as these things form. Questions about that, especially the difference between what makes this retro and why this is different and particularly nasty? Yeah. Oh, yeah. Let me say this really. So I want to be really careful. So what she asks us a question is, when these cells divide, it's passed on to the daughter cells. Does that mean it's passed to your offspring? Not through the germline, because in order for me to pass HIV to my kids through their chromosomes, then the HIV would have to infect the sperm-producing cells. So then it get into the genome of my sperm-producing cells. So when I produce sperm, it would have HIV built in. The way you pass it to your offspring is it's actually in your bodily fluids. So it's not, you don't inherit HIV because the cells which give rise to eggs and sperm are not infectable by the HIV virus. They don't have the right receptors. You can't actually pass it to your kids through their chromosomes. You pass it in body fluids. All right. So let's do the clicker question, and we will call it a day.